continuation of our lecture, we will learn that there are actually two types of culture media for Mycobacterium. So we have usually the liquid and the solid. So the purpose of the liquid medium is for enrichment. Yes, Mary Grace, do you have a question? Okay, so the purpose of the liquid medium is for enrichment, and the purpose of the solid medium, it serves as the primary plate medium. Okay, so what do you mean by enrichment? Ila, um, Ilalagay muna sa enrichment yung sputum para dumami siya. So usually, kapag naging turbid or kapag nagkaroon ng growth, that's the time that we will place it in a solid medium. In solid medium, it will take about 2 to 3 weeks or even up to 1 month before it can grow. So, ganun katagal mag-grow ng Mycobacterium. And there are two types of solid medium. Okay, so solid medium could either be egg-based and agar-based. So, usually, itong mga middle group, we are using this for sensitivity testing. But, kadalasan na ginagamit na primary plate medium for the final cultivation of Mycobacterium would be the egg-based medium. Okay, so we have the Lowenstein-Jensen, Petragnani, American Thoracic Society, and Dorset egg medium. So these are examples of the egg-based medium. Okay, so again, what are the ingredients of this particular solid medium? So in cases of LJ media, kasi this one is the most popular, pag sinabi talagang LJ media, talagang the first thing that should come into your mind should be a Mycobacterium. So it has egg, salt, glycerol, potato, and flour. So as you can see, no, this makes the medium enriched. At hindi nabanggit dito, the medium also contains malachite green. Because malachite green serves as the inhibitory substance. Called the middle group medium, it contains many factors, but one of the most worthy to mention would be the oleic acid. ATS is almost the same as LJ, except that it doesn't have salt, while LJ would have salt. Okay, so that basically would be the difference. Okay, so this is the appearance of the LJ medium. So you can have it in the tube as a slant, but most of the time it is at the plate. So take note that the colonies of the LJ appears like cauliflower-like colonies. And this is the Middlebrook agar. Okay. So all media contains malachite green. So what's the purpose of malachite green? The purpose of malachite green is it serves as the inhibitory substance so that it will inhibit the growth of gram-positive bacteria. Okay? So there are other modifications of culture media. So sometimes they are using the paddle and liquid media. So paddle and liquid media is 2-in-1. Okay? Because it has a Middlebrook agar on one side and LJ or chocolate agar on the other side. And the reason why we're including chocolate agar is that it can detect some fastidious species aside from Mycobacterium tuberculosis, such as the Mycobacterium marinum and Mycobacterium haemophilum. So this is an example of the Middlebrook, septic check, I'm sorry. Septic check. So, ang maganda rito sa septic check, I'm sorry, it's not just 2-in-1. It's actually 3-in-1. Kasi you have the liquid medium here, You have, let's say, for example, a chocolate agar on the other side, and then you have Middlebrook on the other side. So it's 3 in 1. So usually, kapag nagkaroon ng turbidity rito, it means that there is a growth, and that's the time that they place it here. Okay, so that is the example of the septic check panel system. Okay, here is another example of isolator lysis centrifugation system. This one is very good because it has a gel, and usually we place the sputum here, and then we centrifuge the tube so that the substance or the sediments will be the one. We will be using the sediment for culturing and for direct microscopic examination. Okay, so as you can see, there are several composition of the mycobacterial culture media, but noteworthy to mention would be the presence of additional substances because they are really fastidious. So culture media, perhaps this is just one of the few culture media that I've seen that have included fresh whole egg. So kayo nakakita, culture medium that has fresh whole egg, okay, as part of the uh, substance. Kaya nga siya tinawag na egg-based media. So pag nakita kayong egg-based media, so this include American Thoracic Society, Lowenstein-Jensen, and Pregnani. And then we also have here the graph, and then the Mycobacterium, and then Middlebrook is agar-based. So as you can see, the Middlebrook doesn't have any egg at all. So that's why it's called agar-based. Okay? So, among the Middlebrook, pag sinabing 7H9, this one is broth-based. So, pag sinabi natin broth-based, it's actually liquid. It's a liquid medium. And if it's liquid, it's being used for enrichment. Okay? So, level of laboratory services. This is according to the American standard, but the principle, of course, may be applicable only. So this is defined by the American Thoracic Society. Okay. So extent of service. Okay. It could be specimen collection only. When you say specimen collection only, that laboratory would only be collecting specimen. But we say acid-fast stain or inoculation or both only, that laboratory is a reference laboratory, then they can actually do the acid-fast staining. But the third is isolation and definitive identification of Mycobacterium tuberculosis, meaning to say that laboratory is already considered to be as reference laboratory, and that reference laboratory may be capable of identifying Mycobacterium tuberculosis up to the species level. And definitive identification means that aside from identifying Mycobacterium, that laboratory would also assist in selection of therapy by a selection of therapy with or without drug susceptibility testing. Okay? So, therefore, according to these categories or extent of services, the American Thoracic Society can categorize laboratories as to level 1, 2, and 3. So if it's level 1, that lab can only collect specimen, but they cannot perform any mycobacteriological procedures, and they have to send it out to another laboratory. Level 2 means that they can perform acid-fast staining, that's microscopy, and then they can also isolate and identify sometimes and perform susceptibility testing for Mycobacterium tuberculosis. Level 3 means that they can perform everything: microscopy, isolation, identity, and perform susceptibility testing, not just for Mycobacterium tuberculosis, but for all species of Mycobacterium tuberculosis. Okay, so that is the extent of the level of services. These are the categories of the level of services according to the extent of their capabilities of what they can actually. Okay, but in the Philippines, I guess our level of service would be at most at level 2 only. Although there could be some lab that can perform level 3, but most of the time it is at level 2. Level 1 is usually designated for perhaps for the health center, but that is actually not almost always applicable. So take note that Mycobacterium tuberculosis is a slow grower. It will take about 10 to 20 days at 37 degrees centigrade for you to have a visible culture or growth. Okay, so they have small, dry, scaly, warty colonies, buff in color, and they are cauliflower. So they are described to be as cauliflower. So there are two types of growth. Okay, eugonic. The first type of growth is eugonic, and the second type of growth is dysgonic. Okay, so when we say eugonic growth, it is a luxuriant growth. So marami talaga kayo makikitang colonies when we say eugonic growth. And most importantly, eugonic growth is being enhanced by glycerol. Okay? Pag sinabi naman natin dysgonic, the growth is scanty, smooth. So pag sinabing scanty, it is less abundant as compared to the eugonic growth. So to prove my point, this is an example of eugonic growth. So you'll be able to see the luxuriant growth of Mycobacterium. Okay, Mycobacterium tuberculosis. Okay, liquid media. So again, I did mention liquid media a while ago, and I told you that liquid media is an enrichment. Okay, and after growth in liquid media, it must be inoculated in solid media because inoculation in the solid media is the gold standard. Okay, so how long should it stay in the liquid medium before inoculating in the gold standard? So the answer is more than 37 weeks. More than 6 weeks at 37 degrees, except for septic check. Because for septic check, you have to wait for up to 8 weeks. So there are many commercially available liquid media. MGIT, Mycobacterium growth indicator tube. So that's what we meant by MGIT. Positive result is fluorescence. Bactec, it is actually automated, semi-automated medium. And growth index is being shown in the screen. Mag-alarm yung machine kapag nagkaroon na ng growth. Septic check, you have to invert it regularly, and turbidity is the presence of growth. And then we also have the continuous monitoring system, and then, as what I mentioned, the Middlebrook 7H9. So this is the MGIT medium. So if there's fluorescence, it means that there is growth. Then you have to inoculate it in the solid medium. So this is Vapid Check. Okay, Vapid Check is actually or Bactec medium. This is being used for blood and culture and Mycobacterium. So you place the tube here, okay, and then if there's growth, it will alarm, and the LED indicator at the door will tell you which particular slot has actually the growth. So after it has alarm, then you can now place it in the solid medium. But if after 6 weeks there's no growth, then you can just report it to the physician, no growth. Okay, so that is the beauty of the Bactec medium. Okay, Middlebrook 7H9. This is an example of the visible growth of Mycobacterium tuberculosis. So the turbidity is equivalent at this particular So it's kind of difficult, no? Mas maganda pa rin yung Bactec because you don't have to actually rely on your eyes to check if there's growth or not. Okay, so how do we identify Mycobacterium? First of all, you have to remember that Mycobacterium is an acid-fast organism. Okay, and there are two general colony morphology. Okay, And they could either be smooth and soft, and the other one is rough. But if we're talking about Mycobacterium tuberculosis, they are rough kasi nga ang ating description, they are warty and appears like cauliflower. Warty and appears like cauliflower. Okay, so they have this dense, but they have this fried egg appearance with dense center. Particularly, Mycobacterium tuberculosis is actually characterized by cording. If you will be looking at the microscope, it will appear like the strands. Okay, so the bacilli appears like a strand. So we call it the, I think I mentioned it to you last week. We call it the cord factor. We call it the cord factor, and the cord factor serves as the virulence factor. And if you could remember my lecture last week, the chemical that is responsible for the cord factor is known as the 5' trehalose. This is the sugar that is responsible for that. Mycobacterium intracellular, on the other hand, would have a fried egg appearance, okay, without the cord factor, because the cord factor is only seen in virulent strain of Mycobacterium tuberculosis. Okay, growth rate. If it's less than 7 days, we're looking for the rapid growers. Okay, so usually rapid growers are non-pathogenic, but slow growers are mostly pathogenic. So such as the Mycobacterium tuberculosis, Bovis, and Avium complex. So the temperature may sometimes help narrow the species. So usually we standardize it at 35 to 37 degrees centigrade. Okay, so let's talk about photoreactivity. Okay, photoreactivity can be classified into 3. Okay, so some Mycobacterium is capable of producing yellow pigment. And this particular yellow pigment is due to carotene. Okay, so the first category that we have here is called photochromogens, meaning to say they would only produce carotene pigment when exposed to light. So that's why we call them photochromogens. And then the second category is called scotochromogens, meaning to say they would produce a pale to orange color with or without light. So with or without light, they would produce a yellow to orange color. And then the third one is non-chromogenic. So when we say non-chromogenic, they will not produce any pigment at all. They will not produce any pigment at all. So they are buff or they lack color. Okay, sorry for that. Okay, so these are the categories of Mycobacterium. Okay, so meaning to say, in order for us to categorize them, first is that you have to identify first the growth rate. You have to identify first the growth rate. Okay? And then, after identifying the growth rate, there is what you call pigmentation. Okay, so after identifying the growth rate, you have to identify the pigmentation. So, if it's buff or if it's orange. So, kapag nag-orange na siya in the presence of light, we are looking at photochromogens. But if it's buff, okay, after exposure to light, in the absence of light pala to ha, in the absence of light, kapag nag-orange siya, photochromogens yan. Kasi ang photochromogens, magproproduce siya ng ano, sorry, hindi pala to, ang photochromogens will only produce in the presence of light. Ito pala is called scotochromogens. Okay? Tapos, pigmentation after exposure to light, this is yung photochromogens. Kasi nagkaroon ng pigmentation after exposure to light. Nagets ba ninyo? In the absence of light, nagkaroon ng pigmentation, that is scotochromogens. Okay? Tapos nagkaroon ng light, then that is photochromogens. Okay? Without exposure to light, tapos na-expose na sa light, hindi na-expose sa light, wala pa rin color, this is a non-chromogens. Okay? So, this is the three categories according to pigment production. Okay? So, ngayon, after classifying them as scotochromogens, photochromogens, and non-chromogens, the next process is to perform biochemical testing. So, ito na mga different kinds of biochemical testing which I will be discussing later on. Okay? So, ito naman sa slow grower. Okay? So, slow grower naman, slow grower is actually very crucial because kapag slow grower, we are mostly pertaining to pathogenic Mycobacterium. Okay, pathogenic Mycobacterium. So, for example, if the growth rate is between 3 to 7 days on LJ at 37 degrees, so ibig sabihin in 3 to 7 days nag-grow na siya, 3 to 7 days nag-grow na siya, okay, so that is considered to be as particularly a slow grower, pero at the same time, hindi pa rin siya, wala pa rin siya sa categories ng mga Mycobacterium tuberculosis. Because usually, Mycobacterium tuberculosis will take about 2 to 3 weeks before you can see visible growth. Okay, so these are the possible biochemical tests so that we'll be able to identify this particular species of Mycobacterium. Okay, so tuberculin test, ano ba yung tuberculin testing? Tuberculin testing is a skin testing. Okay, so it is a skin testing and it utilizes the principle of delayed hypersensitivity reaction. Why did I say so? Because it will take more than 24 hours before you can see the result. In the old days, they are using the old tuberculin, which is the considered to be considered as a mixture of filtrated glycerol broth culture concentrated by evaporation on a water bath. But nowadays, we are using the PPD or the purified protein derivative. It is prepared from culture filtrates of bacilli, grown on synthetic medium, and purified by precipitation with PCA. Okay, so this is the PPD. Okay, so why is it important? It is being placed in our skin and then we measure redness. Kasi pag nagkaroon ng redness or erythema, it means that we develop hypersensitivity reaction to Mycobacterium. And it means that you are already exposed to Mycobacterium tuberculosis. Okay? So, there are several ways of doing it, but the most popular one is the MAN2 test because it is through intracutaneous injection of tuberculin syringe. And then von Perke, we have to rub it into the scratch in the skin. And then the Volmer patch, you apply to a clean area into the skin on a piece of gauze on tape. And then the Moro percutaneous test, we are including it together with the lalolin ointment, and then the time test, a drop of antigen on skin surface, and injecting it with the use of a multi-prong inoculation. So that is a time test. Okay? So this is an example of the tuberculin syringe. So you actually in, inject the PPD or purified protein derivative, and then you measure the erythema after 24 hours. If it's more than 10, it's more than 10, it means that you have previous infection, exposure, or or you have been immunized with BCG or Bacillus calmette. Within 5 to 10, that's indeterminate. And then if less than 5 millimeter, it means that you are not yet exposed to to tuberculosis. Okay? So this is the von Perkey. So an example of the von Perkey. Volmer patch, ipapatch lang yung lalolin ointment. And then the time test using a multi-prong. So feel ko pinakamasakit yung time test. Okay? So let's talk about biochemical identification. Okay? Um, for biochemical identification, why is it important? As what I told you in our algorithm, biochemical identification will actually help us confirm whether we're looking at Mycobacterium tuberculosis or not. So one is niacin accumulation. So most mycobacteria can convert niacin to ribonucleotide. Mycobacterium tuberculosis will convert it to nicotinic acid, and it will produce cyanogen bromide and aniline. And a positive reaction is a yellow color. Okay? So Mycobacterium tuberculosis is unable to convert niacin to niacin ribonucleotide kasi kino-convert lang niya sa nicotinic acid. Sorry. Okay. So and then that's why Mycobacterium tuberculosis, since they are unable to convert, they are considered to be negative. So niacin reacts with cyanogen bromide in the presence of primary amine or aniline to form a yellow compound. So this one is a positive reaction. Nitrate test. So the principle of nitrate test is some bacteria would have nitroreductase enzyme, which will allow them to convert nitrate to nitrite. And a positive test is Mycobacterium kansasi and Mycobacterium tuberculosis because they have the enzyme nitroreductase. Okay? And the presence of nitrite is detected by the presence of sulfanilamide and naphthyl ethylene diamine or diamine reagent, which is characterized by the presence of a red diazonium dye. So this is a highly positive reaction in nitrate reduction. So if you are asked during the exam what reagent would be responsible for this, this is called the sulfanilamide and naphthyl ethylene diamine reagent. Okay, catalase. I think I have already introduced to you catalase, but for Mycobacterium testing, there's a twist because you have to add twin AT detergent since because of the presence of the mycolic acid. Again, enzyme splits hydrogen peroxide into water and oxygen. So the very fact that oxygen is present, a positive reaction would be the presence of bubbles. Okay. And since this particular one is actually different from your slide catalase, it's being done in a column, you have to measure the height of the column up to 45 millimeter. So yung bubbles dapat more than 45 millimeter to be considered as positive after 5 minutes. Yan. So, hindi siya yung slide method. And this one is more than 4.5 because of the addition of the twin AT. So, iba yung iba yung catalase test ng Staphylococcus kaysa dun sa catalase test dito sa Mycobacterium. Because in sa Staphylococcus, we usually do it in slide, but here we are doing it in test tube. And we measure the height of the column. Ito lang may measure natin, 4.5 millimeters. Okay. Aryl sulfatase test. Principle, Mycobacterium fortini chelonate complex secretes aryl sulfatase, and this particular enzyme can split 3-phenolphthalein from tripotassium salt of phenolphthalein disulfate. So the enzyme aryl sulfatase can split because they have the ability to hydrolyze the bond between the sulfate and the aromatic ring structure. So phenolphthalein salt is the main component of the reagent, and you have to use it in oleic acid medium or oleic acid agar. So all you have to do is add a small amount of alkaline solution in the form of sodium carbonate onto the surface and observe it onto the surface of a 3-day-old culture. So it has to be specific. The culture has to be at least 3-day-old. And if purple color becomes apparent, then that is considered to be as a positive reaction in aryl sulfatase. So here we're looking at Mycobacterium forticium chelonate complex. And then twin AT hydrolysis. Okay, so some bacteria, some Mycobacterium, okay, possess a lipase that can split twin AT. So twin AT therefore serves as what? Substrate of lipase. Diba pag sinabi natin lipase, or pag sinabi natin substrate, it is a substance being acted upon by the enzyme. Okay? So some Mycobacterium such as Kansasi, Gordonii, Marinum, Lavescens, Gastriterrae, and Triviale, Malmoense, they have lipase that can split twin AT. So it modifies the optical characteristic of the test solution from straw yellow to pink. So pag naging pink to, then that is considered to be positive for twin AT. And then iron uptake is for Mycobacterium forticium. So Mycobacterium forticium is able to take up iron from inorganic iron-containing reagent. So yung reagent na yon is actually made up of ferric ammonia citrate and develops a rusty brown color after prolonged incubation in the presence of citrate. So ito. So pag nagkaroon ng rusty brown color, that is considered to be as a positive. So without iron uptake, it is blue-green, but pag nagkaroon ng rusty brown color, it means that it has already converted ferric ammonium citrate to iron oxide. Okay, so that is iron uptake. And then the pyrazinamide test will hydrolyze pyrazinamide to pyrazinol acid and ammonia. So a presence of yellow-brown color is considered to be as a positive reaction. And then tellurite reduction test will reduce potassium tellurite to tellurite black color. So blackening of the bottom is considered to be as a positive reaction. Urease test based on the principle that some Mycobacterium can produce the enzyme urease, which can split ammonia, and it is characterized by a deep pink color. So that is a urease test. So we also have inhibitory test. So when you say inhibitory test, we add something into the medium, and that particular substance can inhibit the growth of a certain Mycobacterium species. For example, NAP can inhibit Mycobacterium tuberculosis complex. PCH will differentiate Mycobacterium bovis from tuberculosis because Mycobacterium tuberculosis is susceptible, meaning to say there is zone of inhibition and Mycobacterium tuberculosis is inhibited, whereas Mycobacterium bovis is resistant. Also, sodium chloride tolerance will inhibit most Mycobacteria because they cannot tolerate high salt concentration except for Mycobacterium flavescens and triviale because they have the ability to grow even in the presence of high salt concentration. And then McConkie agar. Usually, Mycobacterium tuberculosis and other Mycobacterium species cannot grow, okay, on Macaque. But Mycobacterium chelonate fortuitum chelonate complex can grow on Macaque agar without crystal violet, because crystal violet is an inhibitory substance. And growth inhibition by T2H or paraffin 2 carbolic acid hydroside. Okay, so Mycobacterium, the T2H will actually inhibit the growth of Mycobacterium bovis, and they can grow in the medium containing this compound. Okay, so ibig sabihin kapag meron T2H yung medium, most of the Mycobacterium will grow except for Mycobacterium bovis. So that's paraffin 2 carbolic acid hydroside. And then HPLC is another technique in chemistry laboratory. And the reason why HPLC is also applicable because of the mycolic acid. So mycolic acid, again, is actually found in the cell wall of Mycobacterium. And this is the reason why we call them acid fast. It is a long chain of fatty acid. We need to extract first mycolic acid. Okay? So that we can determine whether the organism has mycolic acid. And then, of course, nowadays, hybridization and nucleic acid amplification test, or PCR, is also being done. And this one is highly specific, but it needs training. But you know what? In the lab, we now have a commercially available kit. Yung nga yung pinakwento ko sa inyo, yung gene expert. It will even tell you whether you have isolated an XDR or MDR Mycobacterium tuberculosis. Okay. So susceptibility testing is difficult because it requires experience and trained staff. So there are several methods such as the proportion, absolute concentration, and radioisometric method. Okay. So these are for inhibitory or susceptibility testing. This would be the ideal concentration of anti-TB drugs. So as you can see, no, you only need a very small amount of isoniazid. But isoniazid can be of two concentration, isang 0.2 tsaka isang 1.0. Streptomycin can also be of two concentration, 2.0 and 10.0. Rifampin, 1.0 lang, and ethambutol is 5.0. This is the baseline concentration of anti-TB drugs. So aside from these drugs, we can also use the ethionamide, capreomycin, cycloserine, kanamycin, and pyrazinamide. Okay? So where are we using this? In 7H10 medium. So I told you talaga, yung Middlebrook is usually being used for susceptibility or sensitivity testing. Skin test, immunodiagnosis can be done through skin testing, which I've already explained to you, yung tuberculin testing, and also through serology. Okay, so let's talk about the complex of Mycobacterium. So remember that there are three major complexes. Okay, the Mycobacterium avium intracellular strophyllation complex, the tubercle bacillus complex, and then the atypical mycobacteria, which can also be known as pseudotubercle, unclassified, anonymous, or MOTT, or non-tuberculous mycobacteria. Parang ayaw nilang isali talaga to. Okay. Pero ang pinaka-clinically significant is ito. But for AIDS patient, this one is also significant, the MAIS complex. Okay, so, and then Runyon's classification, which I have already discussed to you. They classified the Mycobacterium. Itong atypical Mycobacterium, they are further classified using Runyon's classification. So I have explained this to you already, yung photochromogens, production of yellow pigment in the presence of light, scotochromogens, they will produce yellow pigment whether in darkness or in light, and non-chromogens will not produce any yellow pigment either in dark or in light, and then we have the rapid growers. Okay, so so these are examples of non-chromogens. So non-chromogens include Mycobacterium tuberculosis, Avium intracellulare, bovis. So ito talaga yung first three na talaga namang highly pathogenic. And the rest are actually not really that common. Yung Celatum, Gastre, Genavensis, Hemophilum, Malmoense, Terrae, and Ulcerans. Itong ulcerans, pwede siya mag-cause ng skin ulcer and can also be seen among AIDS patients. And then the rapid growers are the Chalonnae and Fortuitum. Okay, so under the photochromogens, we have the slow growers. Ito yung Asiaticum, Kansasi, Marinum, Simiae. And the scotochromogen slow growers are Gordonese, Zulgase, Crocolaceum, and Xenophi. And the rapid growers photochromogens are Flay and Smegmatis. But don't worry, usually, madami siya masyado, but usually, most of the, in the board exams, ang concentration natin are in the Mycobacterium tuberculosis. So talagang kadalasan yung question ay nandoon. So we have now come to the second part of acid-fast bacilli. So this time, we'll discuss Mycobacterium leprae, lepra cells. So when you say lepra cells, acid-fast bacilli are found inside the epithelial cells and monocytes. Okay. So Mycobacterium leprae is also known as the Hansen's bacillus. So usually, you'll see them arranged in cigar packet, and we cannot cultivate them. They can only be cultured at the footpad of armadillo. Okay, so they have the ability to oxidize a neurotransmitter DOPA, and they have predilection towards peripheral nerves. So kadalasan, kapag merong leprosy ang isang tao, nagiging manhid siya, they cannot feel anything because the peripheral nerves are destroyed. There is also glycine in the cell wall. So this is the Christian Hansen. So he was the one who discovered Mycobacterium. So the GC contents of DNA is 55.8 lower than other AFP. GC is the DNA, di ba, yung GC content, yung yung cytosine and guanine. Okay? So it's actually 55.8%. So halos hindi sila mag-anak ng Mycobacterium tuberculosis. Ang common denominator lang nila is meron talaga silang microlic acid, which is assigned as C20. Okay? The specific antigen is phenolic glycolipid, and pyridine is the one that would be responsible for the loss of acid fastness. So this is the armadillo. Imagine, no, kung gusto mo mag-cultivate ng Mycobacterium leprae, manghuhuli ka ng armadillo at ilalagay mo sa food pan. Okay. So this is the Hansen's bacillus. So again, yung Hansen's bacillus is also characterized by red. Oo nga, kasi nga, they are acid-fast bacilli. Okay. There are actually two types of leprosy. Okay, so we have the tuberculoid type and the other one is the lepromatous type. So take a look at the incubation period. Sobrang tagal niya. But if you will be asking me which one is a lesser evil, it is the tuberculoid type. Kasi let's compare ha. Yung tuberculoid type is anesthetic type. So ibig sabihin magmamanhid ka lang dito. But yung lepromatous type is the nodular type. Pag sinabi kasi natin nodular type, there is an extensive tissue destruction with disfiguring lesion. So magdi-disfigure yung face mo, yung pag minsan without any joke, yung earlobe mo matanggal. And if you have lepromatous leprosy, dapat palagi kang nakamedyas. Kasi otherwise, yung part of your foot habang naglalakad ka, pwede siyang matanggal din. Okay? So, unlike yung tuberculoid, there's a few erythematous or hypopigmentation flakes with flat centers, raised demarcated borders. Few AFB, low infectivity, and this one is positive for Lepramine test. Yung lepromatous naman has diffuse nerve involvement with patchy sensory loss, but there are many AFB and high infectivity, but it is negative with Lepramine test. So ito nga yung tuberculoid, very few patches lang, unlike yung lepromatous. So it's another example of lepromatous leprosy, no? Kita nyo talaga na magkakaroon ng disfigurement. Okay, nodular type. So that's the reason why they call it nodular type. Okay, so Lepramine test, it's the same thing as the tuberculin, except of course we're using a different, we're not using PPT. Unlike in tuberculin testing, we're using PPT. I say the same because same principle. still uses the principle of delayed hypersensitivity reaction. So, two types of reaction. If reaction is seen within 24 to 48 hours, we call it the early or the Fernandez reaction. If the reaction is seen after 3 to 4 weeks, we call it the late or the Mitsuda reaction. Okay, treatment. Meron bang treatment? Yes. Multiple drug therapy. Okay, in the form of dapsone, which is the diamino diphenysulfone, and the combination of rifampin and clofazimine. Prevention is also through this. Okay, so that ends our discussion on acid-fast bacilli. Thank you very much for listening. Okay, so